Good morning, students of standard 12th. Today we are going to study topic gel electrophoresis from chapter 11 biotechnology. Gel electrophoresis. It is a technique for separating DNA fragments based on their size. Students, gel electrophoresis is a very important process in which the DNA fragments, that is DNA pieces, based on their size, that is based on their length, is separated. Students, this technique in which the DNA pieces are separated based on their size forms one of the most important steps of DNA fingerprinting, in which a very huge length of DNA is cut into small pieces of DNA, and further these short pieces of DNA are separated by the help of electric field provided on the agarose gel. So students, various steps of the electrophoresis you can see here: the first, the second, the third, the fourth, the fifth and the sixth. Where in the first process, the big length of the DNA you can see here is being cut into small pieces, which is further kept on the agarose gel. You can see here the gel. Third, it is being separated by the help of electric field. Fourth, with the help of nitrocellulose paper, it is being separated. And finally, it is presented for research work. Initially, DNA is being cut into fragments by restriction endonuclease. Students, as I said earlier, that the first stage of the gel electrophoresis is that the bigger length of the DNA, actually students, these are the DNA, bigger length of the DNA is being cut, that is, has to be cut into smaller pieces by the help of a specific enzyme called as restriction endonuclease. Students, we have studied very well in the earlier classes that restriction endonuclease is the specific enzyme which has the capability to cut the DNA at proper sites. If the restriction endonuclease cuts the DNA 1000 base pairs apart from its attachment, then that is called as type 1. But if the restriction endonuclease has the capability to attach the DNA at a specific site and cut the DNA into two at the same site, then that is the restriction endonuclease type 2. And if the restriction endonuclease enzyme is able to cut the DNA at 26 base pairs away from its attachment, then that is called as restriction endonuclease type 3. So students, we have already studied about the three different types of restriction endonuclease and their function. And the same function of the restriction endonuclease here is being applied, where you can notice the big length of the DNA is being cut by the molecular scissor restriction enzyme type 2 into small fragments of the DNA, which is mentioned as restriction fragments because these are the fragments which has been produced by the action of restriction type 2 enzyme. DNA is further being DNA is further being negatively charged can be isolated with the help of electric field. Students, generally we know that DNA is negatively charged, and if the DNA is negatively charged, it is quite obvious that it will be attracted towards the positive charge. So students, in this electrophoresis tank, you can notice that the small pieces of the DNA has been kept. These small pieces of the DNA which are negatively charged will definitely be attracted towards the positive charge provided in this electrical appliance. In this electrical appliance, the two plates emit different charges. So DNA being negatively charged gets attracted towards the positively charged and starts getting drained. When this drain of the negatively charged DNA towards the positively charged electrode of this electrical appliance is very important, because here during the drift from the negative electrode to the positive electrode, the small DNA fragments get arranged according to their length. Agarose gel is a specific matrix which is used to isolate DNA fragments kept in electric field. Students, it is the agarose gel which is the specific matrix of fluid which is used to isolate or separate the DNA fragments which are kept in the electric field. So students, we can say that the presence of agarose gel helps the small fragments of the DNA to drift quite easily to the anode of the electric appliance and further get arranged according to their size. Which also can be clarified with this that gel with fragment fractions by size. So the gel which is there with the fragment gets further differentiated into groups in the form of size. Further, the separated DNA fragments are treated with a chemical name ethidium bromide, followed by exposure of UV radiation. So students, after the collection of the small DNA fragments towards the anode region of the electrical appliances, the separated DNA fragment is further treated with a specific chemical ethidium bromide. Students, here ethidium bromide helps the DNA to get recognized quite easily, which further is exposed to the UV radiation, which further amplifies the recognition of the smaller fragments of the DNA. So students, here in this picture you can easily notice that the small DNA fragment has got arranged according to their size, which is further hybridized, that is which is further added with the DNA probe, that is the ethidium bromide. Students, here ethidium bromide is the DNA probe because DNA probe is that specific sequence of the DNA which gets sticked to the another DNA with the same specific nucleotide sequence. So students, we can say DNA probe actually works like a sticker which has been designed in such a way that it can stick to a specific place only, and due to which that specific place where the DNA probe sticks to the DNA can be easily studied. After hybridizing the small fragments of DNA with ethidium bromide, which is the DNA probe, it is further exposed to the X-rays, which further amplifies the recognition of the small sizes of the DNA. Now students, let's discuss a very important question concerning the topic. What is the advantage of gel electrophoresis in DNA fingerprinting? Gel electrophoresis helps in the isolation of DNA based on their size, which is further hybridized with radioactive DNA probe. So students, we can easily understand the importance of gel electrophoresis in DNA fingerprinting, where it is only the gel electrophoresis which has the capability to isolate the different fragments of DNA based on their size, which later on can be hybridized with DNA probe.